And welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is shift 2 question paper and I am discussing the part C section. In this section I will be discussing the questions from molecular biology. This was conducted in the year March 2025. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR Net JRF Life Science being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, see for CSIR Net exam and you will find them there. So coming to the first question, to study different DNA double strand break repair pathways, a construct is developed that contains a neomycin selectable marker gene flanked by two inactive GFP genes. Okay, so this is the neomycin resistant genes and it is flanked on both sides by inactive GFP. One is disrupted GFP and the other is truncated GFP. So it will not give any color or it will not give any fluorescence. The first one is inactivated by insertion of ISCE1 recognition sequence. So this is the ISCE1 site. So the truncated GFP, it is from 100th base pair to 730 base pair. The disrupted one is having the full GFP sequence, but only around 350 or somewhere near 400 or something like that. There is an ISCE recognition sequence being included and it is inactivated. The other one has a 99 base pair deletion at the 5' end of the gene, that is over here. The induction of the ISCE1 endonuclease will create a double strand break in the first GFP sequence. So ISCE1, it will create a double strand break in the this GFP sequence. Now what are the expected outcomes? So they have given something like gene conversion pathway, then single strand annealing pathway and non-homologous end joining pathway, which are the various pathways that are involved in double-stranded break repair mechanism. We will see each one of them, then you will be able to understand the question and answer it. So the double-stranded break repair mechanism usually involves a homologous recombination pathway as well as the non-homologous end joining pathway. And homologous recombination pathway, it involves the single strand break repair pathway as well as the not single strand break repair pathway, sorry, synthesis dependent strand annealing, that is SDSA, that is synthesis dependent strand annealing and double strand break repair pathway. So these are the two pathways that are involved in homologous recombination method of repairing the double strand break. So this is your DNA, you are damaging the DNA and it resulting in double strand break. Now in homologous recombination, the double strand break are repaired by using a homologous DNA template from a sister chromatid. Okay, so you use a homologous DNA template, DNA as the template for repairing this double stranded break. And this homologous recombination, it is usually active during the S phase and the G2 phase of the cell cycle, because at this stage sister chromatids are available. Now the first step in both the pathways which are involved in homologous recombination is, that is SDSA and double strand break repair pathway. The first step will be end resection. Okay, now what is this end resection? That is, the 5' end of the broken DNA are degraded, leaving 3' overhangs. So this is the 5' end, this is 3' end. This is your DNA, this is 3' end, this is 5' end, this is 5' end and this is 3' end of the DNA. Now this after break, you can see one 5' end over here, 3' end over here. So this 5' end, it will be degraded off, and you will get a structure like this with a 3' overhang. See here. Similarly, this region of the second strand will be degraded off. That is your 5' end, leaving a 3' overhang over here. Now what does this 3' overhang end will do is, it will invade the homologous DNA duplex. So this is your homologous DNA, the red one which I have drawn here, that is your homologous DNA duplex. It will invade the homologous DNA duplex, resulting in a D loop, and it will use that as the template for the synthesis of DNA at the broken site. Okay. Now after this, what happens in single in SDS, that is synthesis dependent strand annealing method, is the invading DNA, that is this DNA, that is your green one with the double strand break, it will be displaced and it will anneal, and this region, that is the after synthesis, it will anneal to this end of the that is the end at the other side of the double stranded DNA break. So this region, it will go and anneal over here. That is what you see here. This is the single strand break after repairing the second strand of the DNA. This region has not been repaired. It will also follow the same technique. Okay. So here it will be annealed to the other end of the, or the overhang at the other side of the double strand break. The homologous DNA, it will be retained without any crossing over. So here what do you get? After DNA synthesis and ligation, you will get a non-crossover product. That is what is happening in SDSA mechanism of repair. Now what is happening in double strand break repair? Here also the same steps are taking place: double strand break, end resection, strand invasion, DNA synthesis. Now here one 3' end of the DNA invades the homologous DNA homologous DNA duplex, displacing one strand and forming the D loop. Now this second, this one, the second 3' single strand DNA end, or this overhang, can also invade the same homologous DNA duplex, or it can invade another homologous DNA duplex, and it can result in the formation of what is called as homologous holiday junction. Okay. So that is what is written here: second end capture, DNA synthesis and ligation. So here also all these can result in holiday junction. Now this holiday junction, it will undergo resolution and can result either in crossover product or non-crossover product. Because it is the strand invasion is, or I can say the homologous sister chromatid is being used as a template, so chances of crossing over is there. Crossing over means exchange of genetic material between the broken chromosome and the template homologous chromosome, and it can result in genetic diversity. So this is homologous recombination method of strand repair, and it is error prone mechanism. Okay, that you have to remember, it is an error prone mechanism. Now what is this gene conversion? Now gene conversion, it can occur through both SDSA and double strand break repair mechanism, and it will also result in either crossover products or non-crossover products. Okay. So gene conversion, it is also an error-prone mechanism of DNA repair. Now coming to the next mechanism, that is single strand annealing or SSA. SSA is an other mechanism for repairing double-stranded breaks in DNA, and here also it will utilize homologous sequences flanking the break for repair. That is, it is not using a homologous chromosome. Here it is using homologous sequences on the same DNA molecule flanking the break. Okay, that is being used. In homologous recombination method, it is using the homologous DNA from the sister chromatid. Here sister chromatid is not involved. The same DNA strand is involved. Now what is it doing in a single strand annealing technique? So this is your homologous sequence. This blue ones, I hope you can see that. And the red one, that is the region of the break. Okay. Now what will happen is, here also there is resection. So where the 5' nucleotides are removed, creating single stranded ends, 3' overhangs, okay, or single stranded ends. Now these two homologous region, that is one over here and the other over here, they will come together, and as a result of that, this red ones, they will be displaced off or they will be removed off, and these two will form complementary base pairs. Okay. So what do you get? You get a shortened DNA in the end because this flanking region is removed. Okay, see here, this is removed, whereas the blue ones have formed complementary base pair. So this was the initial length of your DNA. Now it became shortened. Why? Because these bases have been removed. Okay, so the intervening sequences, I will say it is called as intervening sequences of the DNA has been removed. So that is it with the single strand annealing technique, and this is also an error prone mechanism. Okay, so because these sequences are deleted from the original DNA strand, so it is an error prone mechanism. Then comes the non-homologous end joining mechanism. So here also it involves the direct ligation of the broken ends. Here also no homologous template is involved, no homologous sister chromatid is being involved. So it is a direct ligation of the double-stranded breaks. So there is some enzymes like KU70, 80, DNA PKs. They are the different enzymes that are involved in the non-homologous end joining. Then you have ligase 4 and XRC4, CC4 that is involved in the ligation after repair. Then first will be the end binding, the binding to the ends of the double-stranded break. Then there will be end processing, that is it involves either the removal of certain nucleotides to fill the gaps or it can lead to small insertions or deletions at this repair site. So because there is involvement of insertions and deletions while during this end processing mechanism in the non-homologous end joining repair mechanism, so this is an error prone mechanism of double-stranded break repair. So the one which is not an error prone mechanism is your gene conversion. The other two are error prone mechanism. Now come back to the question. So here this region of the GFP is disrupted by introduction of an ISCE site, and this is your truncated GFP. This is your neomycin resistance marker gene. Now this site can be repaired, but this cannot be repaired. It is already truncated GFP. But here it can be repaired, and because this has led to the introduction of a double-stranded break in over here. Now what are the outcomes they have asked? If the double-stranded break is repaired by gene conversion pathway, yes, cells will be GFP positive and neomycin resistant. Yes. So gene conversion pathway, it is just like your homologous recombination method, and it is error. It is not error prone. It doesn't result in any errors. So the repair is perfect, and it will result in the repair of this disrupted GFP, and the GFP will start to function properly. So it will be GFP positive, and the neomycin resistance gene is not harmed as a result of this repair. So neomycin it will be neomycin resistant also. So A is correct. Now if double-stranded break is repaired by the GC pathway, okay, GC is gene conversion, cells will be GFP positive and neomycin sensitive. No, they will be neomycin resistant. So B is wrong, A is correct. So 2 gone. Now CC, if the double-stranded break is repaired by single-strand annealing pathway, cells will be GFP positive and neomycin resistant. No, they will be GFP negative and neomycin sensitive. I told you in a single strand annealing pathway, it uses the homologous region in the same DNA strand, or I can say it will utilize the homologous sequences flanking the break to mediate its repair. So here if you see this, you can see that it is over here, the break is there, and if this portion, that is around 350 to 400, is disrupted, that is somewhere over here, this region, 1 minute, that is around this portion is the one which is undergoing the break. So it is similar to this over here. It is the same region in this sequence. Okay, now this flanking sequence, that is somewhere over here, this flanking sequence, and over here, that is on either side of the break, will be homologous to this flanking sequence over here. Okay, so they too will result in the formation of complementary base pairing, and the intervening sequence, that is from here to here, this portion, the one in green I have marked, it will be removed. Okay, the flanking sequence will be removed, and they too will come together, resulting in formation of complementary base pairs and repair of the base. That is what is happening in single strand annealing pathway. So what do you think? Will the cells be GFP positive? No, this portion is deleted. Neomycin is not there at all, so they will be neomycin sensitive. So that will be the answer for C will be wrong. Okay. If double-stranded break is repaired by single-strand annealing pathway, cells will be GFP negative and neomycin sensitive. So C is not correct. That makes option 1 gone, option 3 gone. So what is left out? A and D, that will be the answer. Okay, what is it that they are telling in D? If double-stranded break is repaired by non-homologous end joining pathway, it is an error-prone pathway. Cells will be GFP negative. Of course, that is correct. And neomycin resistant. Yes, that is correct. So D is the answer. And non-homologous end joining, it is not disrupting your neomycin resistance gene in any way. Only near the break, some sequences will be either added or deleted. So it is resulting in small mutations near the disrupted site, near the break. So it will be GFP negative but neomycin resistant. So D is the answer. Then next one, in a study, researchers replaced the natural promoter of a gene with a synthetic promoter that contains point mutation in the Tata box. Okay, now the real promoter, that is your Tata box, which was natural, they have replaced it, and in place of that a synthetic promoter which is having a point mutation in the Tata box was introduced. Now because of this point mutation, the Tata box will not be able to bind to Tata binding proteins. So the following outcomes could result from this modification. Okay, which of them are incorrect statement? Oh fine, they have asked for incorrect statement. Okay, now see the picture, I will tell you. So this is your mRNA promoters which are responsible for the synthesis of mRNA. So you have got the Tata box over here. Then there is the BRE region, that is TF2B recognition element. Then this is the initiator and the downstream promoter elements. Okay. So if there is a mutation in the Tata box, there are other promoter sequences available which can bind to, 1 minute. Yeah. Okay, now there are now if the Tata box is mutated, there are other promoters, that is BRE is there, INR is there, downstream promoter elements are there, and they can result in transcription of mRNA, but the efficiency will be very less if Tata box was there. Okay, if Tata box is there, 100% efficiency, but if it is from the transcription is from BRE, INR and DP, then it will be less when compared to the presence of Tata box or the non-mutated or the wild type Tata box. Now we will see the options. An mRNA will be generated with an alternate reading frame. How is it that possible? Because here the mutation is in the Tata box and it is preventing the binding of Tata binding proteins. The reading frame is not affected. If it has to be an alternate reading frame, then it must be because of some frame shift mutation. But the coding region is not affected, then how it will result in an alternate reading frame? Only the Tata box region is affected. So A is not correct. So they have asked for an incorrect statement. So A will be one answer. See here, they have asked for the incorrect statement. Then the mRNA will be transcribed by RNA polymerase 1 instead of RNA polymerase 2. How is it possible? The point mutation is in the Tata box and it will not cause RNA polymerase to transcribe mRNA. RNA polymerase 1 is responsible for the synthesis of rRNA, and RNA polymerase 1 it relies on different promoter elements and transcription factors to initiate transcription and not on the Tata box. Tata box is primarily for RNA polymerase 2 which is involved in mRNA transcription. So B is incorrect statement. Then transcription may occur with reduced frequency, that I already explained. The gene will be transcribed by RNA polymerase but with reduced efficiency, because it can even start from other promoter elements like INR, downstream promoter elements, and BRE. So the efficiency will be reduced. So that is not the correct answer because it is a correct statement. Then transcription may occur but will always result in the formation of non-functional mRNA. See, if it has to result in a non-functional mRNA, then the gene's coding sequence have to be mutated. Only then it will result in non-functional mRNA. Here the Tata box is the one which is mutated, not the gene's coding sequence. So the mRNA produced will be functional, but the mRNA will be produced with reduced efficiency. That is the only problem. Not with there is no harm with functionality. So D is also correct because they have asked for the incorrect statement. So A, B, D, yeah. Option 2 is the right answer. Then before coming to the next question, I will tell you some responses in bacteria when there is some changes in the environment or stress like that, or when there is a scarce amount of amino acids present. Okay, so there is the first one that is called as the stringent response in bacteria. Now what is the stringent response? So stringent response is seen in cases where amino acids are scarce. So when amino acids are scarce, tRNA will become uncharged. That is tRNA will not be able to bind to amino acids. So they will remain uncharged. But these uncharged tRNA, they can bind to the A site of the ribosome. And this binding, it will result in stalling or stopping of the ribosome. And this stalled ribosome, they will trigger the or activate REL-A protein, that is a ribosome associated protein, and it will result in the synthesis of PPP, GPP, or I can say it is an alarmone, ALAMORNE. It is called alarmone, ALARMONE. And this thing, it will bind to the beta subunit of the RNA polymerase, and it will alter the RNA polymerase specificity, promoter specificity, and it will alter the RNA polymerase's promoter specificity. And because of this alteration in the promoter specificity of RNA polymerase, it can transcribe some genes or it can reduce the transcription of some genes. Okay, so that is what is happening in stringent response in bacteria. So the stringent response, it will allow the bacteria to adapt to amino acid starvation by slowing down growth or by conserving some resources within the bacteria. Now similar to this, there is another response that is by cis-acting elements or cis-acting regulators. Okay, now what are these cis-acting elements or cis-acting regulators in bacteria? So these also allow the bacteria to adapt to changing environmental conditions such as temperature fluctuations or nutrient availability. And these kind of cis-acting elements, they are directly controlling the initiation of protein synthesis in bacteria. Now cis-acting regulatory elements, they are located on the same mRNA sequence that is involved in the synthesis of a gene. That is the same mRNA sequence which is involved in the synthesis of a particular gene will have this cis-acting element located somewhere near the 5' UTR region. Okay. So these cis-acting elements, they can be some hairpin loop structures, or they can be short open reading frames within the 5' untranslated region or UTR of the mRNA. Now, example of cis-acting regulators, this is the case of RNA thermometers. So, these are involved in temperature sensing, and they produce RNA secondary structures, and they are located at the 5' UTR. So, whenever there is a low temperature condition, the RNA will form a hairpin loop structure and it will bind to the ribosome binding site, preventing the binding of the ribosome and initiation of translation. Okay, that is what is happening at low temperature. But at higher temperatures, that is when conditions are favorable, the RBS or the ribosome binding site will be free and it will bind to ribosome and result in translation of the coding sequence. So that is what is happening with case of RNA thermometers. Now there is another one that is also a cis-acting regulator, that is called as the riboswitches. So the riboswitches, they are also a non-coding RNA and are found at the 5' UTR of this particular mRNA, and it has an aptamer domain, and the aptamer domain, it is highly specific for a ligand. See here, the ligand is bound to the aptamer domain of this particular non-coding RNA at the 5' UTR. And what is it doing? It will sequester the ribosome binding site. See here, this is the RBS, complementary base pairs have formed upon binding of the ligand, and the RBS is not available for the binding of the ribosome, and there will not be any translation. So these are called as riboswitches. Now there is something else that is called as the now this is the cis-acting one, means you have got the trans-acting one also. Okay. So trans-acting regulation is a trans-acting regulation also involves regulation of the mRNA translation, and it is also involved in making the bacteria adapt to changing environmental condition or stress. Here it is a trans-acting, means another mRNA which is not involved with this particular mRNA which is coding the gene protein is controlling this mRNA. That is, some other mRNA is controlling this particular mRNA. That is the one which is required for the gene translation. So it is coding. Some other mRNA from somewhere else is coming and controlling this mRNA. Okay, that is trans-acting. So it is mainly by the sRNA. Now sRNA, they are transcribed independently, and they will regulate the gene expression, and they will bind to the 5' UTR of the target mRNA. The ribosome, and thereby the ribosome binding site is prevented or sequestered, and resulting and it will result in non-availability of the ribosome binding site for the ribosome to bind and initiate translation. So it will result in translation repression. Now in some bacteria there are RNA chaperones, that is HFQ, and they play a role in maintaining the stability of the sRNA, as well as they will help or they will facilitate the sRNA mRNA pairing. Okay. Now come to the question. So translated protein act as a repressor, that is translational feedback. Now translated protein acting as a repressor is usually seen in your operon mechanism. So in this mechanism, what happening is the repressor protein, it will bind to the operator and block the RNA polymerase from binding to the promoter, and thereby result in no transcription translation of the structural gene. So A will be ribosomal protein operon regulation, A2. Oh, great. So A2 is seen only in 4. Happily, if you know that answer, then you need not have to see for the other options. Even if you don't know what is sRNA or ribose switches, you can happily get marks over here if you know A. Okay, now what is this? Production of PPGPP in response to amino acid starvation, which in turn regulate transcription by binding to beta subunit of RNA polymerase. I told you it is a stringent response mechanism in bacteria. So B1. Then regulation of bacterial mRNA translation in cis. I told you it involves RNA thermometers and ribose switches. So ribose switches that bind a ligand, C4. Then regulation of bacterial mRNA translation in trans, that is involving sRNAs or small RNA and chaperone. I told you chaperone like HFQ are involved in certain bacteria for regulation of bacterial mRNA translation in trans and require pairing with mRNA. So D3. So 4 is the answer. Then next one, adding mRNA that encodes a eukaryotic secretory protein to a cell-free translation system initiate protein translation. Of course, cell-free translation system means it will have the ribosomes and the necessary translation machinery will be available, and you can add your mRNA of your interest and see for the protein that is produced by this mRNA. So here what they have done, they have added mRNA and it encodes a eukaryotic secretory protein. So it can be any protein which is secreted by the cell to a cell-free translation system, and it has initiated protein translation. Fine. Signal recognition particle in low concentration and endoplasmic reticulum treated with 1% Triton X100. So first you added the signal recognition particle, then you added endoplasmic reticulum which is treated with 1% Triton X100 were added to the cell-free translation system, and they have asked what are the possible outcomes. So if you know the translocation of the protein into the endoplasmic reticulum lumen, you will be able to answer this question. So how does the protein enter into the endoplasmic reticulum lumen? For that, the protein will be synthesized on the in normal condition, in a normal cell, in vivo condition. Okay, the protein will be synthesized on the ribosomes in the cytoplasm. This is the signal peptide. The signal peptide, it will be recognized by the signal recognition particle. The signal recognition particle will bind to the signal peptide and it will halt the synthesis of protein temporarily. Okay, you have to remember that point. Now what will this SRP do? It will go and bind to the SRP receptor on the endoplasmic reticulum membrane lumen, and this, after binding, the SRP will dissociate off, and the synthesis of the polypeptide will start again, and it will enter into the translocation of the endoplasmic reticulum membrane. The signal peptide also will be entering into it, and once it has, the polypeptide has entered the lumen, the signal peptide will be cleaved off by the signal peptidase, and the protein will be released into the endoplasmic reticulum lumen, and the protein can undergo various foldings into its final conformation. Okay, that is what is happening in endoplasmic reticulum lumen. Now come to the outcomes that they have mentioned. Now before that in the question they have mentioned the endoplasmic reticulum is treated with 1% Triton X100. Now what is this 1% Triton X100 doing? It will disrupt the endoplasmic reticulum membranes, or the endoplasmic reticulum membrane will become solubilized. Now because of that, the SRP receptor which is present on the reticulum membrane will also be gone. Okay. Now what will happen here? The SRP, it will bind to the signal peptide on the newly synthesized protein. Then it will not be able to enter the endoplasmic reticulum lumen because SRP receptor is not there on the endoplasmic reticulum membrane. The endoplasmic reticulum membrane itself is solubilized or it is disrupted. So no SRP receptor available for binding. So once I have already told that once the SRP will bind to the newly synthesized polypeptide, it will halt the protein synthesis. Okay, it will halt the protein synthesis temporarily. Then it will keep searching on the endoplasmic reticulum membrane whether any SRP receptor is available. And it will find that it is not available. So it will not be able to enter the endoplasmic reticulum lumen. So the protein synthesis, wherever it was halted, it will remain halted there. Or you will get a truncated protein or shorter protein. Okay. That is what is happening here. So the answer here will be protein synthesis will begin. Okay, it will begin because they have told initiate protein translation. It will initiate protein translation, but you are adding signal recognition particle. It will bind to the signal peptide and it will halt the protein synthesis. So it will, here there is no signal recognition particle receptor on the endoplasmic reticulum for binding. So it will terminate prematurely, leading to short products. So that will be the outcome. Then next one, below in the table with the list of post-translational modifications on proteins and amino acid residues that are correspondingly modified. So which post-translational modification are correctly matched with the amino acid residues they typically modify? So phosphorylation, it is histidine. Yes, in addition, it is the addition of phosphate group and it occurs mainly on the side chain of serine, threonine, tyrosine, etc. But in some cases it can occur in histidine, lysine, arginine also. So A is correct. Then ubiquitination, it usually occurs on the lysine residues of the target protein. Then it can also occur on the cysteine, serine, threonine as well as on the N terminus of the methionine residue. So B is correct. O-linked glycosylation, it occurs on the amino acid serine and threonine. Asparagine, it is involved in N-linked glycosylation. So C is not correct. Then hydroxylation, if you remember, you have got hydroxyproline. So proline is involved, hydroxyproline in your collagen. Okay. So proline is involved. Then there is hydroxylysine also, that is lysine residue or lysine amino acid is involved, not cysteine. So hydroxylation is also not correct. So A and B are correct. That makes option 4 as your answer. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of CSIR net been discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, see for CSIR net exams and there you will find. Thank you.